Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Quick note before we begin, the Finding Genius Foundation, as part of the Finding Genius podcast, has recently completed a book about understanding viruses. So the creation of this book was to interview 100 virologists, ask them a lot of deep, difficult questions, take the most difficult questions, and then re-interview the top 25 or so and ask them the hardest questions I could think of. And we compiled that all into a book. So you'll see question and 4 or 5 experts' answers. Question, 4 or 5 experts' answers. There's about 30 questions in the book. I think it's a great read for the layperson and for the researcher. It talks about a lot of speculation in the world of viruses, such as are they alive or not, and why is it important? Why do viruses go latent or hidden or ineffective or sit in a person or an animal or another creature for weeks, months, years, and then suddenly become virulent and affect that person? So there's a lot of really provocative questions in the book. It's now on Amazon. So if you go to Amazon and type in Finding Genius, you'll see the book on viruses. It's also on Kindle. The Audible version is in production and should be ready in approximately a month. But if you want to go and order it now, you can do so again by going to Amazon or Kindle, or go to findinggeniusfoundation.org and go to publications. There's an opportunity as well to get the transcripts of all the interviews and to hear the original interviews themselves. If we put them all together, the book would be about 1,000 pages, but we condensed them down to make it juicy and concise and tight and very interesting. So I hope you'll check out the book. We're now working on one about cancer, but this is going to be our goal is 3 times a year to come out with these masterclass books that I think will inspire new scientific research, and I hope you'll check it out. Thank you. Hello, this is Richard Jacobs with the Finding Genius podcast, now a part of the Finding Genius Foundation. I've got a great returning guest, Robert A. Gattenby. He's a professor at the Moffitt Cancer Center. And I'm going to be asking him questions in regards to the cancer book that I'm putting together. You may have heard, if you're a listener, that we did a virus book a few months ago. It's now on Amazon and Kindle. You just type in Finding Genius on Amazon or Kindle and soon to be Audible. You can pick up the virus book. It's got some really provocative, super interesting questions, I think. And our goal here is to do the same with cancer. So, Robert, thanks for coming back.
Thanks for having me.
All right. So to start off, do you believe that cancer is a living entity unto itself, have its own homeostatic drive and essentially, self-preservation agency and all that? Or do you think it's really just a collection of rogue cells that don't work together?
Well, I think that there's, from an evolutionary point of view, each cancer cell is the unit of selection. This is the evolutionary unit of selection, meaning that they act like they're single-cell organisms. However, like other individual organisms, they can act as groups. Herds, for example, each individual within the herd is the unit of selection, but the herd can act in ways that promotes the fitness of each individual member. And these are sometimes called allele effects. In fact, even Darwin notices that as some populations get larger, the usual rules of evolution would be that the proliferation rate, their fitness, goes down because their population is approaching some kind of a limit, in the carrying capacity. But it often goes up. That is to say, the proliferation rate goes up. And this is because individuals can benefit from the presence of others. And sometimes that can be safety, but sometimes there can be better acquisition of resources and so forth. So I think the cancers have that component as well, because each cancer cell is its own entity, but cancer cells also have to act loosely in groups to do what's called niche construction. They have to, for example, promote blood vessels to come in to provide nutrients. They have to break down extracellular matrix, and probably they act together to overcome the immune system. So I guess that's a long answer, but I think there's nothing in cancer that is inconsistent with what goes on in evolution in living systems. There's nothing magic about it. not an evil entity. I don't think it has agency in the sense that it acts in a willful way. In fact, I think specifically does not do that. And so I think it's a straightforward evolutionary process. I think that the cells start as normal and become independent of tissue organization structures of rules, and therefore they begin to proliferate on their own, where their survival, their death, or their proliferation is dependent upon their own properties and not signals from the environment. And that dependence on their own properties is what allows them to evolve.
If normal cells as part of a tissue, though, are essentially working for the, I don't know how to put this, working for the greater good or working for the good of the tissue or of their localized group,
Yes.
What is it about cells then that changes in cancer that makes them independent for a time? And at what point do they rejoin some kind of collective? Like if I have a tumor of, a tiny one of 100 cells, are they acting as one, or it's only at the billion cell stage it acts as one? Like what are your thoughts here as to why is there a difference between normal cells and cancer cells? What is that difference?
Sure. Well, normal cells have the same fitness function as the multicellular organism. Their life and death is dependent on the survival of the organism and of tissues, of signals from the organism. So if the organism, the local tissue tells a normal cell to die, it's time for you to die because I need you to die for whatever reason, the cell dies. And if it says I need you to proliferate, the cell proliferates. So the proliferation, survival, death are all dependent upon these tissue signals, so that the cells are essentially subservient to the kind of this master organization of the multicellular organism. Cancer cells become independent of those signals, and I think they can become independent because they essentially become deaf and blind. They become unable to receive those signals. But the other thing that can happen is the tissue itself cannot give the signals because of age, because of illness, because of trauma, because of infection. So that now the cancer cells are proliferating or dying or surviving based on how they interact with the environment rather than the tissue signals. So they're completely independent of local host controls. And as a result, they have what's called a self-defined fitness function. So they're now out there evolving the same way as any single cell organism in the middle of a pond or under the dirt, in our gardens and so forth. So I think that the cells never really act as a single age, as a single organism, but they can get into these loose areas where they can promote environmental changes that promote the fitness of each member of that community, which again is observed in nature as through what are called aggregation effects, allele effects, and so forth. So I think that the individual cancer cell is always the unit of selection, but there can be these organizational structures that develop within the tumor that simply develop spontaneously because of the need to make blood vessels to fight back the immune cells and so forth.
Yeah, like if I picture a tumor of, let's say, a billion cells, and it's 1 centimeter in diameter, and there starts to emerge, let's say, a hypoxic or an anoxic zone in the middle. The tumor appears to say, all right, let's turn on angiogenesis to get blood flow to the inner part of the tumor. But again, it seems to be acting in concert in some way.
What happens is, of course, that each cell in that region becomes hypoxic, and so it begins to produce VEGF or other angiogenic factors. That's what the cells do. That's their natural response. So now you have a group of cells that are all hypoxic, that are all producing VEGF. Each one of them is doing it because of their local conditions, but in effect, the whole group is now sending lots of VEGF out, and so the signal goes to local blood vessels, and it begins to send sprouts in and so forth. So it's individuals acting at a local individual level, but because you have a group of individuals that are subjected to the same environmental stresses, they're effectively they're acting in concert. So it would look like they're cooperating with each other, and in fact they're simply all doing the same response that sums up. And so since they're all making VEGF, the amount of VEGF that's coming from that region is increased, and it will promote the influx of vessels, but the cells, the vessels that come in are always poorly constructed.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Because as soon as the blood flow to this area goes up, the VEGF is turned off, and there's no evolutionary imperative for them to do what happens in normal tissue next, which is to revise the blood vessels to even the flow to every region in the tissue. There's once the cells have their oxygen, they have no reason, to help out anybody else in the tissue. So there's no cooperation in that sense. So it's a very limited form of sort of group actions.
How do you think cancer first arises? And when it arises, what do you think the first abilities are? Do you think it's unlimited proliferation, or is it immune system evasion? Like, how do you think it first starts?
Well, I'll go with what my own view is that it first has to become the unit of selection. It has to be independent of the local tissue controls, so that its proliferation is dependent on its own fitness, and then it will evolve. So I think that there's probably not one pathway. I think there's probably many pathways to becoming an independent living organism within the single cell organism within the tissue. And so I think it sort of depends on where you start. I guess that my analogy here are cavefish. If even Darwin was aware of cavefish. They live in the total darkness of the cave, and because of that, they lose their eyes and they lose their pigment, and they get sort of exaggerated tactile organs. And for decades, people thought that they were all the same species. And it turns out there's 85 different species that give rise, fish species that give rise to the cave morph. And what happens is that it's convergent evolution. They all become virtually identical. They all develop the same phenotypic responses to the cave, but they do so through all kinds of different genetic pathways, because they all start, each species will come in at a different sort of genetic point, and even different individuals from the same species can come in at different genetic points. So that the evolutionary arc to that convergent phenotype can vary. And so I think that similarly in cancer, certain things, I think there's clearly some things that have to come early, and some things that probably come a bit later. But I don't think that there's a rule that says that this is each step goes in this way. I think it's that kind of convergent evolution that develops to common selection forces.
Okay, all right, makes sense.
Many years ago, I always thought of cancer as like a little ball of cells developing. You start with one or two, and then it's just a ball of cells. But in fact, cancers develop in a very specific geometry. For example, a breast cancer or pancreatic cancer or a prostate cancer develops in a duct. So initially, the duct is has a lumen, and then there's just a layer of epithelial cells around that lumen. Tumors grow into the duct initially, so they're growing into this hollow space. The blood vessels stay on the other side of the basement membrane, so they can't go into the duct. So that these guys are growing into the center of the duct, further and further away from their blood vessels. So there's a distinctive geometry to the beginnings of a cancer. But what it is not is a ball of cells. It's cells developing in these in a circular or cylindrical geometry, or on a spatial on a surface. And so it's not a ball of cells. And I think that we often portray it as that, and I think that's misleading.
Well, what do you think better exemplifies the morphology of it? What is it? actually look like instead of just a ball of cells.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Well, I think what you'll see is cells growing into a duct and forming almost a cylinder of cells. Now, eventually, they have to break through the duct to become a cancer. So those are all what are called in situ cancers. But the early evolutionary dynamics of in situ cancers, meaning before it becomes an invasive cancer, is dominated by the environmental selection forces that are in the duct. And they're imposed because the duct has a specific geometry. And that geometry limits, for example, the blood vessels. They can't penetrate the basement membrane. And typically, you don't see immune cells extensively involved in this. So these are guys that are evolving into that environment. And so then they eventually break through, and that's when they first begin to have to get angiogenesis, and they have to evade the immune system. They have to deal with extracellular matrix. So that's what I mean. I think there's sort of general constraints because the environment in which cancers develop is not static. And again, it's not a ball of cells that just get bigger. If it's in a duct, so that would be called adenocarcinomas that arise in ducts. If they arise in a duct, then the first thing that they do is that they have this cylindrical morphology to deal with, and then afterwards they get this other set of environmental stresses that they have to deal with. So there is a sequence in that sense, but I think otherwise it's pretty much any different way. I mean, you can develop it in a number of different ways.
Well, speaking of I guess I'll call it microenvironment, what would you expect to see in the microenvironment of a primary tumor versus metastases? Because metastases, you have cell type A and environment B, or tissue B.
Yes, it's a very good point. So the cancer cells, let's say we have a breast cancer, those cells have adapted to the breast environment. If they show up in the liver, now they're really faced with a totally different environment, different selection forces, and because they have adapted to the environment of the breast does not mean that they are adapted to the environment of the liver, and so they have to either adapt or die. And if you look at how foreign species move into areas, so they start invading an area. If they come in like birds, they come in from a distance. So you often get multiple different introductions and before one actually sticks and you actually get a population forming. It often depends on the size of the introduction. If you just have a single individual, the chance of that individual proliferating is low. It also depends, of course, on how similar the new environment is to the old environment. Also, whether how adaptive they are, how plastic their phenotype is, and so forth. So, and we're probably very lucky because of those evolutionary dynamics, because it seems, what we know is that metastases are very rare. You can throw thousands, or if not millions, of cells into the blood supply of a mouse, for example, and only get one or two metastases. Once they impact on this foreign soil, most of the time they die. So, and for us, that's very important because we don't get. We know that cancers, large primary cancers, shed millions of cells, and yet metastatic disease is rare. You can find cancer cells in the bone marrow of patients with very localized breast cancer and lung cancer that never turn out to have metastatic disease.
Yeah, well, why do you think metastases are rare, and what, I know there's the seed and soil hypothesis, but why do you think they form and why are they rare?
Well, I think it's exactly that the seed and soil is poorly matched to the time. But whenever you
but why would cancers have tropisms for certain tissues from metastatic sites? It seems like that, right?
It does seem that way, and the answer is I don't really know. But it does. It's clearly not random. So there must be something about the host tissue and the tumor that's developing that is more easily. So, for example, a prostate cancer can more easily adapt to the bone than, let's say, a liver cancer, which rarely goes to the bone. And why, I don't know. I haven't the foggiest idea, but I think it's generally believed that the site of a metastasis is governed in part by the number of cells that go there. So the liver, for example, drains the colon. So a colon cancer is going to send a lot of cells into the liver, and just by statistics alone, it's more likely to find select one that happens, or find one that happens to be able to proliferate in the liver. But it's also then based on that match between the seed and soil. And
what about cancers that metastasize to the brain? They have to get through the blood-brain barrier. It seems like that would be the last place a metastasis would go.
I completely agree. I mean, and again, I think it's fortunate. It's just rare. I mean, the fact that you can have 2 or 3 metastases in the brain, unfortunately for clinically could be fatal, but it may have been billions of cancer cells that passed through the brain, prior to that, and that didn't form metastasis. And it's interesting that, like a glioblastoma, a primary brain cancer, you can find glioblastoma cells in circulation, and yet virtually never does it spread to any other part of the body. So I think I would probably say that seed and soil difference is simply too great to be bridged by the phenotypic heterogeneity that is underlying it. So anyway, I view it as a rare event, but it happens because you have so many events. You have so many cancer cells circulating in the blood that eventually one of them hits by chance alone.
I got you. So even though it's rare, someone with liver cancer, they'll very likely, as time goes on, get metastases with certain tropisms for certain tissues. But even though, again, it's only the occasional cell that you're saying will cause this, there's enough of them that it does happen pretty reliably.
Yes. So in some ways, I think we're very fortunate. We as human beings are fortunate that our cancers are not that effective in forming metastasis. But of course, unfortunately, it does happen enough that typically is what distinguishes a curable cancer from a non-curable cancer, the presence of metastatic disease. So whenever it does happen, it's tragic.
But has anyone done a study of neoplasms versus cancerous tumors, and to see what the differences are? Because you have some of the same behavior, a vast proliferative growth. I mean, has anyone looked at that?
I think that's always been an issue. So benign tumors like a uterine fibroid, for example, can be absolutely enormous. And it can take, clearly those cells proliferated at a wild rate for a while and then stopped. And why did they stop? And the answer is I don't know. And I think that's a really good question. And it probably has not received enough investigation. I think a typical answer would be that it doesn't have the mutations that are required to make it a cancer. It falls short of that for some reason. Another explanation, I think, would be that the host develops a response that ultimately stops growth. And I think either of those or both could be the answer.
How about in a given cancer that has metastasized? Have there been studies where they do like single cell sequencing and attempt to reconstruct in a 3D way the clonal lineages of the variations in a tumor, and also compare primary versus metastases, and look at the the mutations and the heterogeneity, and see if you can see lineage and trace back what formed first and the next and next? Has anyone done that?
Yes, there's been a number of studies with that, and it's not my area, but you can even show, for example, that cells from one metastasis can form another metastasis. So that they're all contributing to this burden of circulating, tumor cells, and you can show that a lot of metastases come preferentially from a single region of the tumor, and so I would say that there's at least a fair understanding of how that process works in general. As always, there are many things that still have to be answered, and sometimes the initial answers to a question don't end up being the final answers to the question. So it's still a work in progress, I think.
if someone looks at a liver tumor versus a pancreatic tumor versus brain tumor, et cetera, are there recurrent themes in terms of structure in each of these types of tumors? Like, if you're looking at liver tumors, do they always tend to have a similar structure? And if so, what is the underlying structure of various tumors?
Well, I think that the pathologist will frequently, can say, well, this looks like a breast cancer, this looks like a liver cancer. They have certain characteristics that are of the cell morphology as well as the, how they cluster together that is typical of these tissue of origin. So they carry that tissue of origin in various ways for most of the life of the cell of cancer. There is what's called a dedifferentiation over time. So a differentiated tumor is one that looks quite a bit like the original tumor. They can form almost duct-like structures, for example, in a breast cancer. They can, squamous cancers can have regions that look a lot like components of the skin. I mean, it's disordered, but at least it sort of recapitulates that organization. And then you typically go from well-differentiated to moderately differentiated to poorly differentiated to undifferentiated. Some tumors never reach that final undifferentiated state. But there is, on the whole, I think, a general progression to become more and more, looking like, single cell, just a mass of single cells that are sort of chaotically interacting, as opposed to these other more differentiated tumors where they do recapitulate imperfectly the structure of the tissue of origin.
Okay, so they do seem to model the tissue they came from, or at least attempt to. Yes,
to me, it's an interesting question about why they do that. So why, if you just said that the individual cell is the evolutionary unit of selection, why do they form pseudoducts? And to me, that's a really good question, and frankly worries me about what's going on there. I think what happens is that there's some evolutionary advantage to that structure there's been research in herds where the phenotypic properties of the members of the herd and how they vary can influence the success of the herd. So where, again, you have an individual unit of selection, but the herd is a sort of group, and there can be some selection on the groups so that the members within the herd are non-random. Now, that's a difficult problem because, again, if each individual cell replicates itself, then that organizational structure can be lost just because they're not proliferating in sync. So I think that, and I suspect that's why you get this transition from sort of well-differentiated to ultimately to, poorly differentiated or undifferentiated. But that's a theoretical.
What about cellular memory? If something was a ductal cell and now it's divided and turned cancerous, it would still retain, I would think, not only the localized signaling from other cells, but it would also retain maybe a cellular memory of its differentiation, its job.
Sure. The thing is that in an evolutionary context, you have to think of what is the benefit that the cell gets from that, or is it strictly, is there something about that its attachment to other cells, even if it's loose? What is gained from that? Because there's a cost to it. It's essentially attached to another cell. Some energy is required for that, but there has to be a gain. And so evolution is this constant optimization process where the cost benefit of everything that the cell is doing is on the table. So it's like, and it's interesting that Darwin, when he thought about the cave fish, he could understand why the eyes would not be used, but he couldn't understand why they would be lost. And literally, cavefish have no eyes. And the answer to that is that there's a cost for the eyes. The cavefish have to invest resources in making eyes, in maintaining eyes, in dealing with maybe the rare infection that comes through an eye and so forth. And so that it's that cost. It has no benefit, but it has a cost. and that means that it has a negative evolutionary selection pressure and will tend to be lost over time. And so I guess what I'm saying is that if the cells of, let's say, a breast cancer are forming pseudoducts so that they are attached to each other in some kind of loose way, that it's not likely that that's just a random event. It's more likely that the cells are getting something from it, and they're optimal. It's not the optimal status of the cells. And so eventually they will tend to evolve so they become totally independent of other cells, but initially that there is some connection that benefits them somehow.
Okay, gotcha. Do you know of anyone that's looked at, let's say, electronic health records for millions of people that have had cancer and see if there's any commonalities in people that develop liver cancer versus pancreatic, et cetera? Has anyone done that?
Well, there's certainly known factors. For example, if you've had hepatitis B or C or D, you're highly likely to get liver cancer. If you have cirrhosis because of alcohol, you're highly likely to get. It greatly increases your risk. Smoking and lung cancer and so forth. For others, where pancreatic cancer is probably a good example. To the best of my knowledge, there have not yet. People have extensively reviewed the clinical history of individuals that get pancreatic cancer and have not yet found some, either inherited trait or something about how people live that can identify a risk. When I first left residency and went to Fox Chase, I attended a lecture, and an epidemiologist presented data that had just been published in the New England Journal, and there was just hysteria in the literature about it. So I remember it very well. And he showed that coffee drinkers tended to get pancreatic cancer, and just hysteria. And over that time now, many studies have come out on pancreatic cancer. And I think it's always been interesting to me that since that time, studies have shown that, it doesn't really change your risk. Drinking coffee does not change your risk. And even some studies have suggested that drinking coffee decreases your risk of pancreatic cancer. So I guess my point is it's really hard to do those kinds of investigations. And so, honing down on a relationship between, lifestyle and many of the cancers has really not been possible up till now.
Yeah, I just figured, there's vast amounts of electronic health data. I wonder if they could find correlations and if anyone looked, but, it's yet another project to do. So,
and there have been a lot of studies. If you look around, the number of studies that have tried to find a lifestyle factor that, results in high risk for, every type of cancer that exists. Some of them have been very successful, smoking and lung cancer, as I said, but most of them have really not. And what you constantly get are these, studies that come out and, they say like broccoli for colon cancer. There's hysteria for about 2 days in the press, and then it goes away. And it turned out that another study doesn't show it. And something as easy as, to me, it should be as easy as, should we be taking aspirin to prevent heart attacks?
Baby aspirin.
Yeah. And those studies are all over the place. So I think it's very difficult to do them. And I've kind of ignored them, to be honest, because they just don't seem to have a consistent story in my reading them anyway.
Well, what parts of cancer do you think need some underlying basic theory to be developed? What's missing in our understanding of cancer? Why haven't we solved it yet? If we're looking at a fundamental level, what do you think is missing?
Yeah, I think that our theory for the carcinogenesis, for the process that makes cancer, but there's a number of reasons that just doesn't fit well with evolutionary theory, for example, and does not fit with some empirical observations, particularly recently. So I think that we've tended to be what has been called cell-centric in these approaches. We tend to view the cancer cell as simply isolated in itself, as opposed to part of a system of tissue and other normal cells, tissue structure and so forth. The other theory I think that's been missing is in treatment. I think that we've really kind of, if there's any theory behind it at all, it's this idea that the more cells you kill, the better that you do. And I think that's been, and this idea of magic bullets, which I think has been around for more than a century, the idea that what you need to do is find an agent that kills cancer cells selectively and leaves normal cells unscathed is the ideal cancer treatment agent, and we have to find something like that. So to me, those are the two that come to mind as in need of theory at the moment.
What role do you think microbiome plays in cancer? The reason I ask is I interviewed a lady named Florencia McAllister who studied pancreatic tumors, and she found that there was a microbiome not only in the pancreas, but the pancreatic tumors had a slightly different microbiome themselves. So, if a cell turns cancerous, I would think its membrane expression of various antigens and ligands changes, and so it would attract different localized microbes that would partner with it and feed it metabolites, et cetera. What do you think?
I mean, I think that's plausible. Again, the problem is that we keep making these observations without any good theory. So if the microbiome affects, let's say, cancer, the development of colon cancer, which would make perfect sense. So then the question is, how does it do it? What are the factors that specifically, draw a line from here to there? How does the change in the microbiome alter the mucosal surfaces that then gives rise to cancer? The problem with microbiome stuff, and I think with things like diet, just what we just talked about, or other lifestyle things, is that human beings are often inconsistent. The microbiome, can change overnight. It can be different in different places in the colon, for example. Just as our diet can change on a day-to-day basis, it can change over a lifetime and so forth. It's hard to track down the effects of something that's always in flux, and to really connect those dots from that to a cancer, because it's likely that if a cancer in colon, for example, develops over a period of 10 years, that polyp, that cancer that's developed, has probably been exposed to a large number of microbiomes over those years. What was their role at these different steps, different stages of the process? And I'm not saying there aren't any. I'm just saying it's very difficult to know that. And I think it's another good example of how we really need, consistent theory to put all these things together.
Yeah, it makes sense. Okay. I was just wondering. In terms of viruses causing cancer, what's the current understanding on how they do that? HPV and some other ones.
Well, I think there clearly are viruses that cause cancer. They are causative of cancer. They HPV being a great example of how the virus gets into the cell, takes over much of the DNA, molecular machinery of the cell, and it does so for the purposes of the virus. But at some point, things can go awry, and you end up getting a cancer cell forming, and therefore cancer. And I think that's a fascinating dynamic. Is really, really interesting. The conventional wisdom now is that perhaps 10 to 20% of cancers are caused by viruses. People that study this sort of thing tend to think that's an underestimate, that it's a much greater number. And I clearly there are times when it's causative. Whether lung cancer, for example, there's everybody knows that smoking is a causative factor, and yet there are also people that have measured HPV that have found HPV genes in lung cancer. So is that part of it or not? And it's interesting. I was at a conference once where someone presented brain cancers, and this viral signature, in all of their patients. And someone in the audience got up and said, we've done that same study, and we have found that signature in none of the patients. So again, difficult experiments to do, but where that there's a clear connection: cervical cancer and HPV and head and neck cancer, and it's unfortunate for the patients, but it's a wonderful laboratory to look at the underlying dynamics that in terms of developing theory for how cancers develop, those dynamics it's important that they be consistent with theory. And I've been involved in this. It's very nice because it's pretty well developed. We know a lot about what goes on there, and it does provide a lot of really good insights into how cancers form.
Okay. I would ask you more about that, but I know that's not really like your area of focus.
And you just did a whole book on viruses, so.
Yeah, but when it comes to cancer, like, a number of questions arise, but, we'll have to table them for a later time. There'll be a viral cancer. There's a whole book to be done on that, I'm sure, with all the interesting things that would come out of it. But
people are using viruses to treat cancer. I mean, it's
yep.
So it's all over the place.
Have you looked into liquid biopsies at all? Do you have any insight or knowledge into what kind of biomarkers liquid biopsies would look at? Do they come from extracellular vesicles? What are your thoughts there?
Yeah, I think that it's again, it's a very promising area of research. I mean, there's a number of things that you could use that for. One is just detection. Can you find something in the blood that tells you that there's a cancer developing, and you need to find it? That would be great. People have thought that there was an infamous paper in the New England Journal, decades ago, that claimed that there's certain proteins in the blood that if you found them, there was a high incidence of cancer that never panned out. And there are various reports that come up, saying that you can predict the imminent development of cancer through various molecular things you can identify in the blood, and that would be great. I think that those are all still, I would say, in development, works in progress. The other thing that which is where I'm kind of more interested in is the use of these molecular diagnostics to understand the intratumoral evolution over time. How does, as you're treating a patient, can you see evidence for developing resistance, and can it be used to give you a sense of how large that resistant population is, and the strategies being used to develop resistance. And
you
again,
mean resistance after chemotherapy, or at what point?
Well, to anything, immunotherapy, targeted therapy, all of those, in any treatment of cancer right now, resistance is the evolution of resistance is typically the biggest barrier to the development of to curative outcomes. I mean, in most cancers now, there's at least one drug that I would say reasonably effective, if not highly effective, and yet in the metastatic setting, and yet cures are rarely seen, and it's because even if you can reduce the population down by 2-3 orders of magnitude, there's always cells that are present that are resistant, and eventually those cells can proliferate and become, the tumor will recur. They're resistant to the treatment, so you have to stop that and go to something else. And usually once that initial strike, once you evolve resistance, each subsequent treatment becomes less and less effective.
Yeah, I guess if you better characterize the heterogeneity of a given tumor or set of tumors, and then you apply the right cocktail of chemotherapy that was really designed to knock out just about all the major groups or types of cancer cells, it might work better than what they're doing now.
Yeah, it's there's a couple of things to think about. One, of course, if you think that perhaps every cubic centimeter of tumor, gram of tumor, has 1 billion cells, then 100-gram tumor, which is not particularly large by oncology standards, has way more cancer cells than there are human beings on Earth. So think about how could you kill every human being on Earth without eradicating all the other populations on Earth, without essentially destroying the planet. And the problem is that it's not just the sheer size, it's the heterogeneity of people. You can see that in pandemics. There's some people that die from a virus, others don't. Some are virtually not symptomatic. And the other thing is that people live in different areas. There's different environments. People live in caves. People live in big cities. People live out in the country. So there's both the phenotypic heterogeneity of a large population, and there's also the diversity of their habitats, of the environment around them. And so think about how could you eradicate that population. Now, if what we've learned in HIV is that if you take 3 drugs or 4 drugs and put them together, each of them targeting different components of the virus, you can, if not eradicate the population, at least knock it down to the point where it becomes unmeasurable for prolonged periods of time. Now, you have to remember, of course, that the HIV virus has, I think, 8 genes. Humans have close to 20,000. So that, taking the lessons from HIV therapy and applying them to human therapy, to human cancers, you have to be careful about. So, for example, you could take, if you have 2 drugs, if you had a 3rd drug and you give all those together, you're still giving it to a population of 100 billion. And so, within such a large number of very heterogeneous cancer cells, the probability, there is a reasonable probability that you will find cells that are resistant to all 3, not just 2. So one of the things that, we've talked about is that, instead of doing it that way, give an initial treatment with, let's say, 1 drug, knock down the tumor. If you can knock the tumor down from, 100 billion cells to, let's say, 1 billion cell, or say 100 million cells, and then give another drug or give 2 or 3 drugs, the probability of finding resistance to that combination of 2 or 3 drugs in 100 million cells is much smaller than 100 billion cells.
Right. Well, what if you were able to determine, if I give a certain chemotherapeutic agent to liver cancer cells, mm-hmm. they'll tend to mutate in such a way as to make them now susceptible to agent B. So, absolutely. Has that been figured out, where they deliberately drive the cancer mm-hmm. down a path where other stuff will affect it?
Yes, and we've done models of that, and we've called them double bind therapy, where and the goal is to and ideally this would be, to me, the ideal way to treat the cancer. You treat, you get an effect, and you also understand the adaptive strategies that are going to evolve in response to your therapy, so that your follow-on therapy then is deliberately made to target that response, and so on and so on, and that would be ideal. It's hard to get drug companies and I guess treating physicians to think that strategically, and so to me that makes perfect sense, but it's in practice simply not done, and I think that it really, we should be considering that. It happens that there was a study that was performed at Moffitt where I think this occurred inadvertently. They took patients with small cell lung cancer who had already been through several treatments. Typically, small cell lung cancers get, at the time they got they start with a platinum-based therapy, and they tend to get a very good response. But then the tumor comes back, and they then get another type of therapy, tumor comes back. So this was now 3rd-line therapy, where they gave them a p53 vaccine. So p53 is a protein that does, among other things, controls repair of DNA breaks, and is often mutated in cancers. And they treated 30-something patients. They got 1 minor response, and so the trial basically failed. But what they then did was they then gave these patients 4th-line chemotherapy. Now, historically, less than 5% of the patients should respond to that drug. And in this case, almost 70% of the patients responded. And the patients that got really good antibodies during the previous trial, that got antibodies against the p53, got the best response. And I think that was a great example. I think if I put my evolutionary hat on, what I would say is that the cancer cells responded to this p53 vaccine by simply reducing expression or just eliminating expression of p53. But then when you throw cytotoxic drugs on them, if you don't have p53 around, you can't repair the DNA toxicity of those drugs, And so, the cells died. And now, to me, that's a perfect example of a double-bind kind of treatment. And to me, it works out perfectly well. But we never do it. I mean, in practice, then, even though I think it demonstrated a way to do it, it didn't. And I guess the sort of tragic story is that the company that made the P53 vaccine went out of business immediately because it was a negative trial. But it's a shame because, in fact, I would say that's not a negative trial. You simply use that, it's sort of a bait and switch kind of thing. You push the tumor to evolve in one direction, and then, it wasn't very effective because the tumor could adapt so quickly. But what you've done is made them weak. You made them susceptible to another type of therapy. To me, that's a great outcome. But in the way that we evaluate drugs these days for cancer treatment, that was a failure, and it would never be approved. So, it's just the way things are, unfortunately.
Well, very good, Robert. We're out of time. I thought we would solve cancer while we're on this call, but we haven't. But I know we've contributed to the knowledge.
We'll try again. Give me another hour, we'll do it.
Just to give you an idea, I looked on PubMed. There's like 4.2 million papers on cancer, and that's just the public ones. That's crazy.
Mm-hmm. Yeah, that's what got me interested in the math, because when I first started to learn about cancer, what you have is an enormous amount of data, but no organization. And in physics, this is the kind of thing that happened with planetary motion, and there's been various times in physics where they just had lots of data and just couldn't fit it together at all, and you couldn't do it until you developed theory that was consistent with. So that's my argument for doing the math. It doesn't. Nobody else believes it, but I'm still in there plugging away at it. So.
Right on. Well, Robert, thank you for coming back. It's been great to talk to you again, and I really appreciate it.
Sure. Nice to talk to you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.